Hello friends, welcome to my channel again. In this lecture we are going to discuss a very important topic in a very easy way. So it is the megasporogenesis and we will discuss about the structure of ovule. So in this lecture we are also going to know very well how the development of embryo sac takes place or the female gametophyte is produced. So we will discuss about the development of embryo sac as well. So development of embryo sac or female gametophyte. So we will discuss about these three things and in a very easy way. So remember the megasporogenesis, structure of ovule, how the ovule is developed and what is its structure, and how the embryo sac will develop. Clear? So remember, before going in detail about this, we will know that the female reproductive part of an angiospermic plant is called gynoecium. So the gynoecium is made up of many pistils, and the pistils are consist of three important structures. So the pistil has three different structures. We know that the stigma and the slender stalk, the style, and at the base it is the bulged ovary. So all these three structures, the stigma, the style, and ovary, makes a single pistil, and it is the female reproductive part. So in gynoecium there may be a single pistil or many pistil. When a single pistil is present, it is called monocarpellary. When a single pistil is present, it is called monocarpellary, and when many pistils are present, it is called multicarpellary. Another two terms are there. When the carpels or the pistils are fused together, it is called syncarpus. And when they are free, it is called apocarpus. So we know about these important terms: the pistil, which makes the gynoecium female reproductive part of an angiospermic plant; the monocarpellary, when the carpel is single or the pistil is single; when more than one pistil is present, it is called multicarpellary. When they are fused together to form a single structure, it is syncarpus, and when they are free, it is called apocarpus. So remember, the pistil has three parts, and the most important part here is the ovary. The ovary has the most important part. It has the most important part, the ovarian cavity. The ovarian cavity is present inside the ovary, which is known as the locule. So the ovarian cavity is present inside the ovary, which has the placenta inside it. So it is also called the locule, and the placenta is present inside the ovarian cavity. It bears the placenta. And the placenta plays very important role in the development of the megasporangia, and the megasporangia is nothing but the ovule. So placenta plays important role in the formation of megasporangia, and the megasporangia is nothing but the ovule. So the ovule is formed inside the ovarian cavity with the help of the placenta. So placenta plays important role. Now remember, ovule is attached with the placenta by a stalk. This is the structure of ovule. So you can see here, I have the structure of ovule here, and the structure of ovule is clearly seen, and the ovule is attached to the placenta with the help of a stalk, and this is the stalk which is known as the funicle. So funicle is the stalk by which the ovule is attached to the placenta. We have studied about the structure of placenta in class 11, and here the funicle is responsible for the attachment of the ovule with the placenta, and the attachment point of the funicle with the ovule is known as the hilum. So it is the hilum that is the point where the stuck or the follicle is attached with the ovule. Now remember, the ovule is covered with one or two coverings known as the integument, which is seen here. Integument is there, and the integument may be one or two. Here we have two integuments. One is the outer and another is the inner integument. The integument surrounds the ovule, and the integument is absent at a point which is known as the micropyle or the micropylar end. So it is the end where the integument is absent. Clear? So remember, the integument is absent at the micropylar region, and opposite to the micropylar end, it is the chalaza. It is the chalaza or chalazal end. It is the chalazal end. Clear? So chalaza is present at the base of the ovule, and micropyle is the opening, is the pore through which the male gamete can enter into the embryo sac. The embryo sac is present at the center. The central position is occupied by the embryo sac. Now remember, nucellus is there, and nucellus is a mass of cell, and it reserves food for the development of embryo sac. For the development of the embryo sac, the nucellus are the mass of cells that present surrounding the embryo sac, and they provide nutrients for the development of embryo sac. So nucellus plays important role in the development of embryo sac. Though remember one thing, there are two integuments in some cases and one integument in some cases. When a single integument is present, it is called unitegmic. When a single integument is present, one integument is present, it is called unitegmic, and when there are two, it is called bitegmic. When there are two, it is called bitegmic. So remember, when a single integument is present surrounding the embryo sac, it is called unitegmic, and when there are two integuments, it is called bitegmic. So it depends on different types of plants. In some cases, in some angiospermic plant, the integument is single, making the unitegmic condition, and when there are two integuments, it is called bitegmic. So here the structure of ovule says that it is the embryo sac is surrounded here in a bitegmic way, means with two different integument. Now we will discuss about the structure of a mature embryo sac, how the mature embryo sac is formed. So first we need to know that the nucellus has certain cells at the apex region. Towards the nucellus there are certain cells known as the archaeosporial initial, and these archaeosporial initial then undergoes certain changes, and a special type of cell is formed from them. It is called the megaspore mother cell. It develops into a megaspore mother cell, which is called MMC. The megaspore mother cell is formed and it undergoes mitotic division now. So the megaspore mother cell, remember, is the cell having a large structure, dense cytoplasm and prominent nucleus. Now it undergoes meiotic division. It undergoes meiosis. And as a result of meiosis, 4 haploid megaspores are produced. 4 haploid megaspores are produced. The 4 haploid megaspores are arranged forming a linear tetrad, forming a linear structure, and out of the 4 megaspores, 4 haploid megaspores, 3 degenerate. 3 of them degenerate, and the remaining 1 enlarge in size, forming the single functional megaspore. So these 3 degenerate and the remaining 1 forms the functional megaspore. The 1 forms functional megaspore. So the functional megaspore is, remember, it is the first cell of the female gametophyte or embryo sac. The functional megaspore. It is very important because from this cell the other cells will produced as a result of mitotic division. So after mitosis, two nuclei are formed and moves to opposite poles. You can see here two nuclei are formed from the functional megaspore as a result of mitotic division. So remember, the megaspore mother cell is having 2n number of chromosome, but haploid, these are the megaspores are haploid having n number of chromosome. So the cells after division from the functional megaspore moves to opposite pole. These are the two nuclei which are resulted from the mitotic division. Then from the 2 there are 4 nuclei. From 4 again due to mitosis 8 nuclei are formed. So remember, these 8 nuclei are arranged inside the embryo sac. The 8 different nuclei are forms 7 different cells. Remember, there are 7 cells but 8 nuclei because the 2 cells synergids on egg cells makes the egg apparatus at the micropylar end. Remember, at the micropylar end there are 3 different cells having 3 different nuclei. 2 synergids, 1 egg cell. They together form the egg apparatus. The synergids are having a some finger-like projections known as the filiform apparatus. The filiform apparatus guides the pollen tube to enter into the synergids. The filiform apparatus receives the male pronuclei that comes through the style and enters into the synergids through the pollen tube. Now the central cell has 2 pronuclei, polar nuclei. In this case there are 2 pronuclei which are arranged like this and forms a single cell because the cytoplasm is fused but the nucleus is separated. So here it is a 2 nucleated structure or 2 nucleated cell. At the chalazal end we have 3 antipodals having 3 different nuclei. So remember, the chalazal end has 3 cells called antipodals and the antipodals present at opposite to the micropylar end, at opposite side of the synergids. Clear? This is all about the structure of embryo sac and how a mature embryo sac is developed in an angiospermic plant. Please prepare note from this lecture, and if you have any doubts, please comment below and let me know. Thank you very much. Please subscribe to my channel, Science Biology.